Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall talk about this programmed cell that is called Parthanatos. Parthanatos is basically poly-ADP ribose polymerase 1 or PARP1 dependent cell death. And this was reported in 2009. Now PAR here stands for the PAR polymer, poly-ADP ribose polymer. Thanatos in Greek means death. Parthanatos is a programmed cell death that we see under physiological conditions. For example, the turnover of ovarian cells in order to balance cell proliferation and elimination. And it's also seen in various pathological situations such as neurodegenerative diseases, cancer, as well as septic shock. Now, one reason we want to talk about this programmed cell death, besides this importance, is that there is some biochemistry here that we have not talked about in previous videos. So this pathway would be very helpful to understand some basic biochemistry that's also very important for cellular function. So one of this biochemical concept is this enzyme called PARP1. So this is a nuclear protein, and this enzyme has several domains. So at the N terminal, we have this zinc fingers DNA binding domains, ZNF1, ZNF2, and ZNF3. And these zinc finger binding domains will bind places in the DNA that are damaged. So any part of the DNA that is damaged, this is the domain of PARP1 that will bind to the damaged site. And then we have the domain called the auto-modification domain, and we'll talk about that in a few seconds. And then we have the enzyme domain, the C-terminus catalytic domain. This is the enzyme part of the PARP1. This is called the CAT domain. This PARP1 enzyme can be activated either by certain signaling pathways or by DNA damage. So what this enzyme does is that it will attach PAR polymers either to itself, which is called auto-modification, or to other proteins, what we call acceptor proteins, and that's called heteromodification. And these acceptor proteins could be proteins such as DNA repair proteins, DNA polymerases, topoisomerases. So these are a lot of nuclear proteins. And by adding PAR to these proteins, it helps recruit these proteins to the damaged DNA site. So besides recruiting important proteins to the DNA damage site, PARP1 also relaxes chromatin in order to facilitate DNA repair. And besides DNA repair, it's also been reported that PARP1 is involved in certain steps in replication as well as transcription. So when there is damaged DNA, what happens is there's a nuclear localization sequence, or NLS, in PARP1, and that sequence helps to direct PARP1 to the nucleus. So the PARP1 enters the nucleus, and then using its zinc finger 1 and 2, it will recognize and bind the DNA damage site. Now zinc finger 3 will now activate the enzyme domain, the CAT domain of PARP1. And once the enzyme is activated, the PARP1 now makes and attaches the PAR polymer to the protein substrate. and that is called par relation. So the par is attached either to the acceptor protein, what we talked about earlier, heteromodification, or the par polymer can be attached to part 1 itself, which is what we call automodification. So that's what this enzyme does. It basically adds par polymer to itself or to other proteins. And here is the reaction for part 1. You can see this enzyme basically cleaves NAD right here. Okay, so that releases this part here, what we call the ADP ribose unit, and this part comes out as the nicotinamide portion, the NAM. This ADP ribose unit then reacts with the carboxyl group of the acceptor protein or PARP1 itself, and that way it attaches the ADP ribose unit to the protein, the substrate protein, via an ester linkage. And after the first unit is attached, the enzyme then uses more NAD and attaches more and more ADP ribose units covalently through glycosidic linkages. So you can see the sugar of this ADP ribose unit is joined to a sugar of another ADP ribose unit through glycosidic linkages, glycosidic bonds. And this forms either a linear polymer, so this can keep on happening, or it can form branches, form branch power polymers. So linear power polymers or branch power polymers. So this is basically what PARP1 catalyzes. It attaches the power polymer to proteins. Now it's important to note that if PARP1 is overactive, that may lead to NAD depletion because NAD is, the polymer is made from NAD. And that may also lead to ATP depletion because NAD synthesis requires ATP. Okay, so that's an important thing to note. Overactive PARP1 can lead to NAD as well as ATP depletion. Another important point to note here is that this PAR polymers don't have to be bound to the protein covalently. Some proteins can bind PAR non-covalently. Okay, so PAR polymers could either be attached to proteins covalently or non-covalently. So with this background, let's now talk about the story about this programmed cell that called Parthanatos. Okay, so we have damaged DNA. So we have damaged DNA in a cell. Let's say first we'll look at what if the damaged DNA is mild. It's a mild damage. Okay, so there is some sort of DNA damage. And that can happen due to many, many different reasons, alkylating agents, oxidative stress, hypoxia, inflammation, including unusual DNA structures such as unpaired DNA. So what happens is when there is DNA that is damaged, PARP1 will come and bind to the damaged DNA. Okay, so it uses its zinc fingers to recognize and bind the damaged DNA. And when this happens, PARP1 is going to change its shape and now become catalytically active. PARP1 now becomes an enzyme. Now, this enzyme will now synthesize PAR polymers, and these PAR polymers will be attached to either itself or to acceptor proteins, DNA such as DNA repair proteins. And these DNA repair proteins will now be recruited towards the damaged DNA site. And in the next step, the DNA repair proteins will go and repair the damaged DNA site. So that's basically what happens if the DNA damage is mild. Now, what if the DNA damage is severe? Well, once again, the PARP now will be activated because the DNA damage is severe. This will be overactivation. So the PARP will now attach PAR, especially branch PAR. Average size of the polymer is about 40 ADP ribose units. And this branch PAR will be attached to the DNA repair proteins, and that will help it recruit to the damaged site and hopefully repair the protein. And of course, this is very severe damage, so chances of repairing is slim. So besides this happening, what we also see is accumulation of the PAR polymer. So in the nucleus, we see a lot of PAR polymer being formed when there is severe DNA damage. In the next step, this PAR polymer will be transferred from the nucleus to the cytoplasm and eventually to the mitochondria. And in the mitochondria, we have this protein here called apoptosis-inducing factor, or AIF. This is located in the intermembrane space, the space between the inner mitochondrial membrane and the outer mitochondrial membrane. This AIF really is a misnomer because it doesn't induce apoptosis. So this AIF actually normally helps maintain mitochondrial function, helps the mitochondria remove free radicals, and also helps in the formation and stability of complex 1 in the electron transport chain. And this binding of this PAR to AIF will release AIF from the mitochondria. So the AIF is released, and it's assumed that this might be helped. This release might also be helped by the fact that a lot of NAD has been depleted. Even enzymes such as proteases such as calpain might be involved in helping release this AIF. Swelling of the mitochondria by the oxidative stress might also help release AIF from the outer membrane into the cytoplasm. Now, once this AIF is released into the cytoplasm, it will now go and bind another protein, a small protein called migration inhibitory factor, or MIF. Now, MIF is a pituitary stress hormone. It regulates corticosteroids as well as endocrine function, and is also involved in inflammation, and it triggers cancer. The next step, this AIF-MIF complex will be transported from the cytoplasm into the nucleus. This might be aided by proteins such as cyclophilin A as well as other proteins. Once the complex enters the nucleus, the MIF portion is actually an endonuclease, so it has nucleus activity, and it will go and break the DNA into large fragments. So this DNA damage, because it's so severe, the chances of repair is very slim. It will now break the DNA into large chunks, large pieces. So besides breaking the DNA, there's also a lot of ATP depletion because, as you know, making these PAR polymers uses NAD, and NAD synthesis requires ATP, so ATP is depleted, and that will lead to the loss of plasma membrane integrity. There is no swelling, but the plasma membrane does get disrupted. So eventually these events the breakage of DNA as well as the increased permeabilization of the membrane will cause cell death. The cell morphology in Parthanatos shares features with both apoptosis as well as necrosis. Then there's a nuclear shrinkage, chromatin condensation like what we see in apoptosis, large-scale DNA fragmentation, membrane disintegration but no swelling. So unlike necrosis, there is no swelling. And there is no apoptotic bodies formed. So this is basically the story of Parthanatos, another programmed cell death. In the next video, we'll look at two other programmed cell deaths. One is called ferroptosis and the other is called cupoptosis. So until next video, thank you very much for watching and we'll see you in the next video.